Okay, good morning, everyone. So we're moving on to the final subsection of this course. So we're still going to be talking about infectious diseases, but we're going to be focusing on evolution of those infectious diseases, which is an area known as evolutionary epidemiology. We're going to be modifying some of the models we've met to understand things like drug resistance or the evolution of virulence. Virulence is the extent to which a pathogen harms its host. We could also look at things like immune evasion as well. So today we're going to go through some concepts about evolution, just to make sure everyone is on the same page when it comes to ideas about evolution, natural selection, what evolution is and isn't. We're going to talk about a variety of different concepts within evolutionary epidemiology, including a concept of trade-offs. And we're going to talk about how we can go about modeling this using ideas about rare mutants invading a population, and then think about competition between those mutants and what's in the population already. Okay, so there's quite a lot of different things to cover today, but I wanted to start off by just going through some general concepts about evolution. The reason is, I think everyone probably, in my experience, feels like they have a reasonably good grasp of what evolution is, but some of the finer details are not necessarily there. Okay, so if I was to ask you, what is evolution? What would you say? Anyone? Any ideas? How would you describe evolution to someone? Yeah?
I guess I would say it's like how a certain species changes over time to become more beneficial.
Okay, so you've got some of the right things in there, some misconceptions as well, but that's common, so don't worry. So this idea of there being some change over time in a species or in a population. So yeah, if I was to describe evolution, I would say it's the change in heritable characteristics of populations over successive generations. So individuals themselves do not evolve. That's why we have this successive generations part. So populations here rather than species, just because the species concept is itself a little bit fuzzy. You might think that the definitions between species are very clear-cut, but when it comes to things like, say, viruses, how do we define what a species is? Bacteria as well, that kind of species concept starts to break down a bit. Populations is generally slightly better. And this heritable bit is key, right? So there's passing on some change from, there's some lineages or descent where change is passed on, and those characteristics will affect evolution somehow. Okay, what is natural selection?
The idea that like the species that happen to have like strong genes will live on start to.
Okay, so you're kind of saying survival of the fittest is what people often say, right? So yeah, kind of like people, I'll talk about this a bit more on the next slide, but people tend to focus mainly on survival because of the idea of survival of the fittest. Really, natural selection is the differential survival and reproduction of individuals due to differences in their characteristics. We put heritable in brackets here. An additional H in here for some reason. Okay. So some common myths. There are many more of these, which you can read here at this website if you're interested. A few things that people often say about evolution or think about evolution. First common thing that I'm sure you've probably all heard, evolution is just a theory. I would say so is gravity. Gravity is just a theory. When we talk about scientific theories, we're talking about a coherent body of both an understanding of the mechanisms at play, so a theoretical basis, as well as empirical observations that support that. So let's say so is gravity. We've seen on the previous slide that there's slightly different definitions for evolution and natural selection. So people often assume that natural selection and evolution are the same things. They are not. For example, you can get evolution by drift. Genetic drift is essentially random effect. So populations can change just because mutations occur. Even if there's no effect of natural selection, they will still accumulate change over time. And those changes are heritable, even if there's no differences in fitness. Evolution maximizes survival or strength, things like that. And this is not, it's not just about survival. If, say, an organism was able to have a really, really long lifespan, was really good at surviving infectious diseases and avoiding predators and all of that, but didn't produce any offspring, it wouldn't be able to pass on its genes. Okay, so it's not all about survival, so reproduction matters. There's a common myth that evolution always results in progress. You said that things are beneficial. This is kind of complicated, but it doesn't always result in progress, or what we might think of progress is not necessarily what evolution does. Evolution has no foresight as well. It can't see some end goal. It acts in a very immediate sense. Okay, so I would say it's a bit of a philosophical question, but what is progress? There's also something that can occur called evolutionary suicide. This is an extreme example of where selection, natural selection, driving evolution can lead to populations going extinct. So even though natural selection is pushing them in a particular direction, they're evolving in a particular direction, that can be bad for, say, the population as a whole. It can be bad for the individuals overall, and it can drive them extinct. Okay, so it's not always a good thing. Or this idea of progress is kind of nebulous. Okay, evolution or natural selection acts for the good of the species. Well, we've just seen above that evolutionary suicide is not a good thing. Also, evolution and natural selection in particular does not act at the what is good at the population level. Really, it's about what is good for individuals or genes. Has anyone heard of The Selfish Gene? Book by Richard Dawkins? No? This is about the idea that genes can be selected for even if they are harmful to their host, say. That's a very brief way of describing it, but it's all about individuals and genes, not what's good for the population. Evolution is random. Evolution is not random, but mutations typically are. Evolution, when there's no selection, can show random effects. But if we have natural selection, then it's not going to be random. Things that are fitter in an evolutionary sense will increase in frequency in a population. Those that are less fit will decrease in frequency. That's fairly deterministic. Evolution is slow. Again, this is not necessarily true. We've seen in the pandemic that COVID-19 or SARS-CoV-2, the virus that causes COVID-19, has evolved very rapidly. It's adapted to us. We've seen different variants of concern coming out, Alpha, Delta, Omicron. That's rapid evolution. This is not taking centuries. This is occurring on a very rapid timescale, on the same timescale as an epidemic, in fact. Mutations are always bad. Nope. If they were always bad, where would we get good traits from? So selection needs variation in order to act. Okay, so there needs to be variation in phenotypes and the characteristics of individuals in a species or in a population for natural selection to act. Some of those mutations will be good, some of those will be bad. So where would we get novel traits from? In an infectious disease context and related today, from a, say, a bacteria's perspective, a mutation that confers a resistance to antibiotics can be good. Okay, more complex organisms have evolved more somehow. And this is not true. People often like to think of humans as being more evolved, whatever that would mean. This idea of, say, like a hierarchy. Part of this is an issue and relates to the point below, where you will see maybe that classic example of a diagram representing evolution, where there's monkeys into man kind of scenario, and there'll be a gradual progression. This idea of progress again, or that we're at some sort of pinnacle level of evolution, that's not true. Related to that, this idea that humans are descended from chimpanzees. We don't. We have a common ancestor. And finally, humans are not currently evolving. Not true. We're still under natural selection. We still experience genetic drift. We are still evolving. Some of those facts are kind of, some of those effects of natural selection are masked a bit because, we have things like medicine that will interfere, and we have social structures that can kind of interfere with evolution, but we are still evolving. Recently, examples of, say, malaria resistance in recent history. In sub-Saharan Africa, there's a higher prevalence of certain genes that are related to malaria resistance. Some of these including things like sickle cell anemia. And that's because natural selection imposed by malaria has selected for genes that are disadvantageous elsewhere, but are advantageous there because of malaria. Okay, so lots of myths about evolution. Maybe you thought you understood evolution before, and you're questioning yourself now. In some ways, that's kind of a good thing. Like I said, in my experience, a lot of people, I think they understand evolution, but don't really understand some of the finer details. Like I said, there are more of these misconceptions here if you're interested. Okay, so what are we going to do today? Today, we're thinking about evolution of pathogens. So pathogens cause infectious diseases. We're going to think about how those pathogens might evolve, and we'll be doing this again for the last two proper lectures on Monday and Wednesday as well. Okay, so what I want to do here is to, first of all, for us to think about if we have, our classic SIR model that we've been looking at a lot. If we've got density-dependent transmission, we have per capita rates of new infections given by beta S. So beta is the transmission rate, S is the proportion or density of individuals who are susceptible. And then we've got a recovery rate in that very simple SIR model. How might we expect natural selection to affect pathogen evolution? So how might it affect these different components that go into that model? Okay, so let's work through this together. So let's think about this new infection term for starters, beta S. So one thing that might happen, remember this beta S is composed of some contact rate and a probability of transmission per contact. So C is your contact rate, and P is your probability of transmission per contact. We might see, for example, P increases maybe. So natural selection might increase the transmissibility of this pathogen, which would increase this value of P here. In principle, it could change this contact rate too. It could increase this contact rate. Why might it do that? Examples of pathogens and parasites that increase or manipulate their hosts in such a way that they can increase onwards transmission. So there's example of pathogen that causes toxoplasmosis in humans. And this parasite, it infects rats and cats. And when it infects rats, it makes them less afraid, and mice as well, I believe. It makes them less afraid of cats. And that's advantageous from the pathogen's perspective, right? Because if they're less afraid of cats, then the cats can eat them, and it passes the parasite on to the cat host. And that completes, it has a complex life cycle that needs to go through multiple host types. So by making the rats less afraid, it can essentially increase this contact rate. It's a more complex scenario than the kinds of things we've been dealing here, because we've got multiple different host types, but it's the same kind of principle. If we think this term here is infected individuals coming into contact as well with susceptible individuals, so perhaps making them asymptomatic might increase this contact rate. So there's potential ways in which selection might affect these different components here. This S, we might not think, that's a host thing, right? Hosts that are susceptible. How does the pathogen evolve to affect that? Well, as a pathogen sweeps through a population, it depletes the pool of susceptibles, right? That's why we get an epidemic. It peaks and then declines. But as we've seen with SARS-CoV-2, if they're able to evolve antigenically, which means that they change their antigens, this can result in immune evasion, which essentially increases that pool of susceptibles again. Okay, so that was the new infection term. We also have this gamma, which is the recovery rate. Because this is a loss term from our infectious class, you might expect there to be selection for lower recovery. In other words, for a longer average infectious period. So that's one way in which they could potentially evolve. What about this second question here? How else might we expect a pathogen to evolve? So what's one of a key thing that pathogens often do to their hosts that's bad, that is missing from our basic model that we've not been talking about at all?
Like death.
Death, exactly. So I'll call this virulence, just because that's the usual way we refer to it. To the extent to which it causes mortality or other types of morbidity, disease. I'll put mortality in brackets here. So its death rate could evolve. What else? If anyone can remember the title of this lecture, that might help. Yes. Great answer. Drug resistance is also a possibility, right? There are various ways in which they could evolve as well, but the thing we're going to focus on here today is this idea of drug resistance. Now, we're going to model this as affecting this recovery rate here. Okay, lots of different ways in which pathogens can evolve. Okay, a little bit of history. So drug resistance is a big problem. We'll refer to this as AMR, or antimicrobial resistance. Yeah, this is being antibiotic resistance as well often. So this is a major public health concern, particularly going forwards. So the antibiotic pipeline, where we discover new antibiotics, and then the timeline between us discovering them and deploying them and resistance evolving is becoming shorter and shorter. So we're running out of antimicrobial treatments for lots of different infections. You may have heard of MRSA, which is a big problem in hospitals. So this is Staphylococcus aureus, a bacteria that is resistant to methicillin, type of antimicrobial. There's also examples of things like MDR-TB, so tuberculosis, that is resistant to multiple drugs, and even something called XDR-TB, which is extensively drug-resistant tuberculosis, and that's resistant to an even wider range of drugs, both first and second line treatments. So this is a big problem because these types of drug resistance pathogens or strains are becoming more common. They're going to make things like routine surgeries become more of an issue. Previously you just, someone goes into surgery to prevent infection, you just give them some antibiotics. If those are not becoming effective anymore, they could get things like MRSA infections. You can't treat them anymore. So this is a big problem going forward. So trying to understand how AMR drug resistance evolves is a big thing. This is where models can be quite informative, help us maybe tailor different interventions or treatments to prevent that from happening. This down here is an example of penicillin. So these are penicillium molds. So I don't know if you know the story of how penicillin was discovered, but it's worth looking at. I'm not going to go into it now, but sort of a serendipitous discovery. Okay. So let's think about some modeling. So one of the key things is to, why aren't all bacteria drug resistant to start with? Why don't they just have this over many, many years? Often in evolution, we see trade-offs. This idea that you can't necessarily be good at everything. Okay, so a particular bacteria may be very resistant to drugs, but that resistance may come at a cost to its growth because it may be having to essentially invest resources or have more genes that slows its replication rates down or whatever. It can't necessarily grow quickly and also be resistant to drugs. It could maybe grow quickly in the absence of drugs or grow slowly in the presence of drugs, but survive. So you can't necessarily do both. And so often we see patterns like this, where say if we were looking at its extent of drug resistance versus its growth rate, say, this is growth rate in the absence of drugs. You maybe get something that looks a little bit like this. So pathogens that are maybe not very resistant can grow very quickly. Those that are more resistant grow slowly, but if there are drugs around, they can survive. So trade-offs are really important in evolution. We have to factor them in whenever we're modeling evolution as well. So when we use antimicrobials to treat people, and this is also a problem in things like farming where they're just used, to prevent infections occurring. So the more we use them, the more we're potentially increasing selection for drug resistance. That doesn't necessarily mean that drug resistance will evolve, of course, but there should be some threshold where as we increase, say, I'll say drug use here, increasing to the right. And if we think of this y-axis as being a measure of evolutionary fitness, so how good a particular species is, we might see something like this. So there's maybe some threshold level of drug use. Below that threshold, it's disadvantageous to be resistant, and above that threshold, it's advantageous. We're not going to go too much into some of the more complex details about this. One of the key components to have evolution is you don't just need selection, you also need variation. Okay, and that variation is generated by mutations. So often we find, this is a bit of an aside, often we find when we're trying to understand, what is the best strategy to treat things, if you don't have very much of, say, a drug, then you have very weak selection. You have lots of mutations being supplied because pathogen numbers are very high, but you might not get selection for drug resistance. So I'll sketch it on here briefly. If we've got something like drug use going left to right here, suppose this red line is our mutation supply. That's related to population size. Okay, so when we've got very low drug use, our population is not impacted. Our population of pathogens is not impacted much. They can replicate at a high level. Whenever they replicate, they mutate. So we can have lots of mutations. If you have lots and lots of drug use, you reduce the pathogen population size, not producing many mutations, since you have a low mutation supply. And then conversely to that, your strength of selection typically looks something like this green line here. So when you've got no drug use, there's no selection for drug resistance. When you've got very high drug use, you've got very high selection for drug resistance. And you find that your rate of adaptation maybe looks something like this purple curve here, where your peak rate of adaptation, your peak rate of evolution and selection for drug resistance occurs at some intermediate, because it has to balance both mutation supply and selection. This was a bit of an aside. I'm not going to test you on that. I just wanted to explain that it's not just about selection, and it's not as simple as saying in this graph that I just drew above, this one here, it's not as simple as saying, I'll just keep your drugs below a certain threshold and everything's fine. It matters about the impacts on mutation supply as well. Okay, let's get back to this though. Okay, suppose we've got a pathogen that follows SIS dynamics, so we've not got any immune class here. We've just got two classes, susceptible and infected. We have density-dependent transmission and recovery occurs when we administer this antimicrobial. So if you don't administer the antimicrobial, there's no recovery. If we have this antimicrobial, then you can get recovery. And we're going to assume that host demographies of births and deaths are negligible over the timescale that we're interested in. And we're going to suppose that our pathogen may experience a mutation that affects its level of drug resistance. We're going to call this level of drug resistance rho. When rho is equal to 0, there's no drug resistance. When rho is equal to 1, there's complete drug resistance. And so it has a multiplicative effect on our recovery rate. So our total recovery rate is now given by gamma, which is our baseline recovery rate, times by 1 minus rho, where rho is this level of resistance. We're also going to assume, though, that this idea of a trade-off, that drug resistance is costly for the pathogen. Obviously, if there was no trade-off, then you would just get selection for complete drug resistance, right? There'd be no disadvantage to this, because it would be, you'd be able to reduce this recovery rate to 0. Individuals, once they become infected, would just always stay infected. So there has to be some kind of trade-off, perhaps. So if we think about our transmission rate, think of this as telling us something about the growth rate within those individuals, or its ability to transmit. And then we think about, not gamma there, sorry, this should be rho, our drug resistance. Maybe there's some relationship between these two things. So the pathogens that have a very high drug resistance are not as transmissible, they can't grow as much, and can't produce as many new transmission stages. Whereas those that don't have any drug resistance or have very low drug resistance can replicate very fast and produce lots of transmission stages. Just remember, we've got this drug resistance going between 0 and 1. Okay, so we can write down some equations to describe this model. Think about our susceptible proportion, the SDT. And previously we had minus beta SI, but now because of this trade-off, we're going to assume that beta is a function of rho. And this is just this graph up here. We don't know what this is yet, but beta is some function of rho. In fact, I'll rename this beta of rho. Okay, so beta is no longer a parameter, a fixed parameter. It's a function of our drug resistance. And then we have the rest of our infection term, proportion susceptible, proportion infected. And because this is an SIS model, when individuals recover, they're going to go back into this S class. And we said that our recovery was going to be gamma, which is our baseline recovery rate, times by 1 minus rho, where rho is our strength of drug resistance. And this is multiplied by I, our infected class. And because we're dealing with proportions here, we've only got two classes, the IDT is just equal to minus the SDT. This is because S plus I is equal to 1. In fact, we could just eliminate either S or I from one of these equations and just have one equation describing everything. Okay. Our R naught in this system, we could calculate it using the methods we've talked about before. Remember, this R naught is the rate at which new infections are produced by one infected individual when everyone else is infected. In other words, this term here, when this S is going to be equal to 1, so everyone's susceptible, the per capita rate, so we ignore the I, so we're just going to have beta times by rho. That's our transmission rate, but we don't have the S there at all because S is equal to 1. So that's the rate at which we produce new infections. And then we multiply this by the average infectious period. The average infectious period is going to be 1 over the loss term. So in our di dt, it's going to be this is going to be our loss term, the rate at which individuals are recovering. So gamma divided by 1, sorry, gamma times by 1 minus rho. So rate at which new infections are produced times by the average infectious period. The average infectious period is going to be 1 over the loss term. Okay, and we can show, but we're not going to do it here. Leave it as an exercise if you want to. We have a stable endemic equilibrium at S is equal to 1 over R naught, and I is equal to 1 minus 1 over R naught. Okay. So to model evolution in this scenario, we're going to make a few simplifying assumptions, and that's going to allow us to write down some new equations and think about how a rare mutation or a rare mutant fares if it tries to invade this population. So we're going to assume that our host and pathogen population are at some endemic equilibrium, which we've just described up here. So they're stable at those levels. And we're going to consider the fate of this rare mutant. And this rare mutant I'm going to call I sub M. That mutant has a slightly different level of drug resistance, rho sub M, and consequently a slightly different transmission rate because beta is a function of rho, so beta of rho sub M. So let's write down an equation for the per capita growth rate of this rare mutant. And this is going to be known as the invasion fitness. So W is going to be a function of rho M and rho. So this is the invasion fitness of a rare mutant that has drug resistance rho sub M in a population defined that already has drug resistance rho. Okay. So per capita growth rates is just going to be 1 over IM divided by DIM dt. So what is this DIM dt? Well, it's going to look very similar to this equation here, but with one crucial difference, which is that we're assuming that because this mutant is rare, so imagine that you've got, a big host population, one individual's in a hospital, say they're infected by this pathogen and they get a mutation within their infection that confers drug resistance. That's not going to suddenly change the prevalence in the population, right? We just want to see, is that going to start to spread or not when it's very rare? And therefore we can assume that our susceptible population stays pretty close to its endemic equilibrium. So in other words, we have our transmission term, or transmission rate beta of rho M. And this gets multiplied by our susceptible proportion, and we're just going to fix this. Okay, eventually, if this invades, it will change this, but we only care about it at the moment when it's rare. So we're just going to, for simplicity, fix this and then see whether it can grow when it's rare or not. If it can grow, then it's going to be able to invade and spread in the population. But if not, then it's going to die out. We don't multiply by IM here because we've got it on the left-hand side here, because we're doing per capita growth rates. And then we have a loss term. So this is recovery, gamma, times by 1 minus rho M. And again, the IM term is going to be on the left-hand side. So all I've done here is I've taken my equation that I had for di dt before. So it would have been this equation here. And I've replaced all my rows with rho sub m's, because this is a mutant, a slightly different trait, slightly different level of drug resistance. And rather than having S, I have S star. So I'm fixing this now rather than letting this vary. Okay, we know what S star is because we worked it out, or we were told what it was up here. It's just 1 over R naught. So we can simplify this and write it as gamma times by beta rho m times by 1 minus rho over beta rho minus brackets 1 minus rho m. All I've done there is I've just substituted in the value of S star, which is 1 over R naught, where R naught is beta of rho over gamma times by 1 minus rho. Okay, so that's just substitution. Okay. Question then is, when can this rare mutant invade our resident population? So remember, this is all about drug resistance. We want to know, when can drug resistance start to spread in our population? So for notational convenience, what we're going to do is we're going to label our basic reproduction numbers for our dominant or resident pathogen in the population as R naught R. So this R stands for resident. And the mutant, which is rare, is going to be R naught with an M in it. So when do we get invasion? Well, invasion is all about when this here is positive. This invasion fitness. This is basically just saying, when it's rare, will our mutant invade or not? Will it grow? Will it spread? So all we need to know is we get invasion when W of rho sub m, rho is positive. If it's negative, then it's going to be less fit than our resident population, so it's going to decline. It's not going to spread. And since we've restricted this rho to be between rho and rho sub m, to be between 0 and 1, that means we can take our fitness that we've just described. We know that these 1 minus rho and 1 minus rho m terms have to be positive. And we need all of this left-hand side to be positive. So when's that going to happen? Well, gamma is just a constant. We can get rid of that. We divide through by gamma. It's a positive constant, doesn't change anything. With a little bit of rearranging, this term here is positive when we get beta of rho m over 1 minus rho m. In fact, I'm going to leave the gamma in there. We don't have to leave it in there, but there's a reason for notation-wise that I'll leave it in there. So beta times by rho m, sorry, beta of rho m divided by gamma times by 1 minus rho m has to be greater than beta of rho over 1 minus rho times by gamma. If these look a little bit familiar, it's because this one on the right-hand side was our R naught that we defined before. And I'm now going to call this the residence R naught. And the one on the left is the R naught, the basic reproduction number for our mutant. So we need the R naught for our mutant to be greater than the R naught for our resident for invasion to occur. This should kind of make some sort of intuitive sense, right? R naught is telling you something about how well a pathogen is able to spread, how well it's able to grow. If our mutant is better able to grow than our resident, then it should be able to spread, right? It should be able to do better. So if R naught M is greater than R naught R, we get invasion. Okay. 2 slides left. This brings us on to something called evolutionary invasion analysis. There's a lot of text here. This is just summarizing what are the key assumptions of the method that we're doing. Remember, we're trying to model evolution. We've just described how a rare mutant can invade. But what we want to know is what happens over the long term, okay? So this particular method focuses on those invasion dynamics, describing what happens over the long term if we assume that we have these three things. So the first thing is that these mutations are sufficiently rare so that our population always reaches its endemic equilibrium before a new mutation arises. So that means that we're not having lots and lots of rapid mutations. We only essentially see one new mutation and then see what happens. The reason we assume that is because everything then boils down to this invasion fitness, okay? We're not having multiple mutants around. We just have one mutant and one resident. So we always end up with this scenario here. We're also going to see that assume that our mutations have relatively small effects, so that the drug resistance of our mutant is very close, but slightly different to the drug resistance of our resident population. This is just so that we can essentially model things as being continuous, you know, so we can treat rho M and rho as being continuous. We're not having big jumps in our drug resistance. And then finally, we're going to assume that if this rare mutant can invade, then it will replace the resident strain and drive it extinct. Again, this means that we don't have multiple mutants in the population or multiple residents. This is known as evolutionary invasion analysis, or something called adaptive dynamics. It's essentially a set of assumptions that allow us to simplify the process of evolution so that we can model it relatively easily. Okay. Why do we do all of that? Well, we want to know what happens not just to one mutant, but what happens to a mutant that can invade. It replaces the resident, becomes the new resident. A new mutant arises. Can it invade? Yes, it replaces the resident. So if you imagine as I'm walking here, this line, this yellow line is describing the drug resistance in the population. New mutant arises, can it invade? Yes. New mutant arises, can it invade? Yes. Okay, so there's a trajectory, and that trajectory is describing how this trait, this drug resistance, is evolving. What we want to know is, am I going to keep going in that direction forever? Is there a point at which I'm going to stop? And how do we know that? We know that by something called the selection gradient, sometimes referred to as the fitness gradient. Okay, I'll refer to this notationally as lowercase s of rho. What this is doing, it's a slightly wordy description here, but it's not actually as bad as it looks. If we take our partial derivative of our invasion fitness, which was W, a function of both our mutant trait and our resident trait, we take our partial derivative with respect to our mutant trait, and then evaluate this whole thing at our resident trait. In other words, set rho sub m to equal to rho. Then this gives us something called the selection gradient. And this is really just telling us how quickly or what direction we're evolving in. It tells you both of those things. Really, we care about the sign of this thing. So this selection gradient, if I turn that word description into mathematical notation, is just the partial derivative of my invasion fitness with respect to my mutant trait. And then we just evaluate this after we've done the partial derivative. We evaluate this by just setting all of those rho m's equal to rho. This comes back to that second assumption from a couple of slides ago, or this slide here, that our mutations have relatively small effects. Okay. So we just want to see if we have a particular trait value, rho. If this is positive, we have a certain level of drug resistance. If this is positive, then that drug resistance is going to be under selection to increase. If this is negative, then it's going to be under selection to decrease, and so rho will decrease. So in our model, what do we have? Well, our selection gradient is just the derivative of our fitness. And our fitness was just, copy it from here, this expression here. Okay. So I'm going to paste this in here. This was our fitness. I want to take our derivative of this with respect to rho m. So what do I need to do? Well, this beta of rho m is now going to be beta prime. And rather than rho m, I'm going to be setting rho m into rho because that's where I'm evaluating it at. So the derivative of this function, this transmission rate, as a function of drug resistance, the 1 minus rho here, that doesn't change. That's just a constant because we're taking a partial derivative with respect to rho m. So that's just coefficient. And likewise, the beta of rho here, that is also just a constant because we're taking a partial derivative with respect to rho m and then setting rho m into rho. So this was the only thing that we took a derivative with respect to, and then evaluated it when rho m is equal to rho. And then this term out here on the right-hand side, we had a minus 1 plus rho m. And then we're taking the derivative of that with respect to rho m. So we're going to have plus 1. So this thing here, we take the derivative with respect to rho m, we would just get plus 1. And so this is our selection gradient. We can also write this slightly neater as, or less, we should write it as beta prime, the derivative of beta, divided by R naught of rho. I'm going to include this rho here just to emphasize that this R naught is a function of rho. Okay. And then we had plus a gamma. So this line, this here to here, is just remembering what our definition of R naught is. R naught is equal to beta times by rho, or beta of rho, sorry, not beta times by rho. Beta of rho divided by gamma times by 1 minus rho. So we're going to have beta prime of rho times by 1 over R naught. That's where this comes from. And then we have a gamma times by 1 here at the end. So, final step here. This selection gradient is telling us both how much we're evolving and the direction in which we're evolving. So is drug resistance increasing or decreasing? If this is positive, then rho is going to be increasing, so drug resistance increases. If this is negative, then rho decreases, and so drug resistance decreases. What happens when this is equal to 0? That means it's neither increasing nor decreasing. This is when we're going to call this a singular strategy. Okay, so when this function here equals 0, then we are not increasing drug resistance or decreasing it. It's constant. So this is referred to as something called a singular strategy, which I refer to as rho star. This is simply the value of rho that satisfies our selection gradient being equal to 0. So again, think about my drug resistance is increasing, it's increasing, it's increasing, and then suddenly I get to a point where it's no longer increasing anymore because the selection gradient has reached 0. If I had been on this side, my selection gradient would have been negative, say, it would have been decreasing, decreasing, decreasing. Now it's equal to 0. So it's say negative on this side, positive on this side, and either way, I evolve to this point here. And that's my singular strategy. And this singular strategy is a little bit more complicated, but we're not going to go into that, at least not today. Maybe we'll do it next week to determine whether it is stable or not, and whether we do actually always evolve to that point. But in this particular model here, this value of rho star will represent an evolutionary endpoint, and this will represent an optimal level of drug resistance for our pathogen. So this type of model can tell us, depending on the relationship between the transmission rate and the level of drug resistance, so that trade-off that we talked about earlier, will give us an optimal level of drug resistance and tell us how that drug resistance will evolve. Okay, the self-study for today is showing exactly what that is in our model. I'll leave that as an exercise. There's been quite a lot of material today. I do appreciate that. I wanted to go through this full example. We're going to be going back and revisiting some of these things next week as well. Okay. That is it for today. I'm holding office hours now if anyone wants to ask follow-up questions or about anything else. If not, have a great weekend. Yes.
So that is a big function.
Beta is a function, yeah.
So how to define this?
Good question. That is something that we would potentially find using data, or it's something that we would define here and think about what do different relationships between the drug resistance and the transmission rate. How does this curve affect where you end up?
So we will define the data function before we do the drug resistant model.
In this particular case, we've just left it as a general function. Okay, so this and the self-study problem here, the self-study problem is asking you to show that this evolutionary endpoint, this singular strategy, its optimal level of drug resistance in this model always satisfies this equation. And you note that this is a derivative of our curve, our trade-off, d beta, d rho. And this is our function itself, rho, beta evaluated at rho star. For any curve, this would be true. So if I go back up to these trade-offs here, this is one particular relationship between these two things. That same equation will hold if my curve looked like this, or if it looked like this, or if it happened to look in the other direction, but that wouldn't really make sense in this context, or if it was a straight line. Any function I use, it would always satisfy that equation. And the exact function that it was would say depend on the type of pathogen. You know, some pathogens might have a very strong relationship, some might have a very weak relationship, and so on. So very good question. This function here depends on, say, the pathogen and the population that we're looking at. Okay. Any other questions? Okay. Have a great week. Or have a great weekend.